Hi everyone, welcome to my channel. If you're enjoying these videos on physiology and biochemistry, please do hit the like button, share it with your friends and subscribe to my channel. Now this video will be dealing with glyoxylate and glycolate metabolism. It is actually a part of metabolism of two carbon compounds. So when we see two carbon compounds, we have acetate metabolism where we have oxidation of ethanol or sugar alcohol oxidation. But here in this video, we'll be mainly focusing on the glyoxylate and glycolate metabolism. Let's first see what is glyoxylate and what is the significance of its metabolism. Now, glyoxylate is a conjugate base that is formed from glyoxylic acid. Glyoxylic acid is a carboxylic acid with two carbons. As you can see over here in this picture, there's one carbon here and another carbon over here. So the two carbon conjugate base of a carboxylic acid is what we call as glyoxylate. Glyoxylate commonly occurs in green leaves in plants, in unripe fruits of many plants, and it is also present in algal species. It is mainly found in the excreta of the algal species, which are growing on forest floors or in the rhizosphere soils. So you mainly find it around plant species. There are also some bodies called as glyoxysomes. Glyoxysomes are they are microbodies which are found inside plant cell, which is very rich in fat. So where it can take part in beta oxidation of fatty acids. I have a different video on that. But those microbodies, those structures are called as glyoxysomes. So they take part not only in beta oxidation but also perform glyoxylate cycle, which we'll talk about a little later. So in other words, I can say that glyoxysomes are actually peroxisomes which contain the enzymes of the glyoxylate pathway along with other regular peroxisome enzymes like catalases and oxidases. So these glyoxysomes are found mainly in all groups of eukaryotes like the yeast, fungi, protozoa, plants. But the glyoxylate cycle as such is not having much significance in animals and including in human beings. So it now currently there is a lot of research which says that no, this glyoxylate cycle is also found in animal cells. But then majorly if you see, there is evidence to prove that glyoxylate cycle occurs in human beings and in other animal cells. Now let's see what is the significance of the glyoxylate metabolism. What happens when we break down the glyoxylate? How does that help the cell, or why would the cell go about in the utilization of this two carbon compound? First of all, it helps to convert the fats in the cell into carbohydrates. So during the glyoxylate cycle, we'll be seeing later, the fat is converted into carbohydrate by the formation of fatty acid, which is then, undergoes beta oxidation to form acetyl-CoA, which then gives us succinate, and then succinate gets into carbohydrate synthesis. So very important to convert the reserves of fat into carbohydrates. This is very much evident, or this is very significant in germinating seeds. So during seed germination, where the plant does not have the option of photosynthesis, seeds don't show photosynthesis, but they have a lot of stored lipids. So those stored lipids in the seeds are converted into carbohydrates by this process of glyoxylate cycle. So this is the cycle, this is the pathway that it takes up. This anaplerotic pathway is what the plant takes up or the seed takes up to form carbohydrates for energy production. We also know that this is the cycle. Glyoxylate cycle is the one which helps us to utilize the two carbon compounds whenever there is an absence of glucose. See, when there is glucose in the medium, the cell would definitely prefer glucose. But in absence of glucose, even two carbon compounds can be used as a carbon source, is shown by the glyoxylate cycle. So this is a very important cycle for mainly for the metabolism of two carbon compounds. And remember that it is a variant of the tricarboxylic acid cycle. We'll talk about that a little later. And it is being performed in the peroxisomes if it is a eukaryotic cell. Of course, in bacteria, or in other prokaryotic microorganisms, you will be finding the enzymes for glyoxylate cycle in the cytoplasm. The next compound that we have in the metabolism of two carbon compounds is glycolate. Notice the difference in spelling. The earlier compound we spoke about was glyoxylate. This is glycolate. You can in fact even say that glycolate is glyoxylated related two carbon substrate. So the common thing between the two is that even this is a two carbon compound. Like you can see over here, there are two carbons that are attached to oxygen groups, and the difference here is that glycolate is actively synthesized in the chloroplast. So in plants, during photorespiration. We'll not be getting into the details of this, but during photorespiration, the plant synthesizes glycolate. Photorespiration is actually one of the major carbon metabolism pathways that we find in plants and other photosynthetic organisms. What happens here is that the very important enzyme called rubisco, that is ribulose 1,5-bisphosphate carboxylate oxygenase, the most abundant protein enzyme on earth, or the enzyme on earth. So rubisco, instead of reacting with carbon dioxide and producing oxygen during the regular process of photosynthesis, goes for another alternate pathway where rubisco actually catalyzes the reaction with oxygen instead of carbon dioxide. So during photorespiration, the plant is taking in oxygen, and this is a wasteful pathway, because then we have, instead of carbon dioxide fixation, carbon dioxide is actually being released over here. There's a lot of energy loss that's happening. That is what we call as photorespiration. Now, during photorespiration, glycolate is being synthesized. So 2-phosphoglycolate, that's written as 2-PG, is converted to glycolate in the chloroplast stroma, and that glycolate is then subsequently transported to the peroxisome, like you can see over here, goes to the peroxisomes to be broken down and oxidized to glyoxylate. So the process where it is being synthesized happens in the chloroplast by the process of photorespiration, but the metabolism, the breakdown, happens with in the presence of, or in the peroxisome, where in the presence of enzyme glycolate oxidase, it is being converted into glyoxylate, as you can see over here. So it is transported to the peroxisome where it is getting oxidized. Then that glyoxylate is then further used, as we will see a bit later. Now this glycolate oxidase is the enzyme that catalyzes the conversion of this glycolate into glyoxylate, and this is a very important enzyme. Actually, glycolate oxidase is also found in human beings, but then and it is very important in kidney stone formation. But the main point to remember here is that glycolate is actually a glyocylate related 2-carbon substrate. It is a 2-carbon compound. It gets form glyoxylate. So we earlier spoke about glyoxylate that comes from glycolate. Where did glycolate come from? Glycolate came as a product of photorespiration in the chloroplast. So the breakdown of glycolate happens in the peroxisome, and this is what we are going to focus about. Now, where does this glycolate help us, or glycolate breakdown help us? It eventually helps us in the formation of amino acids. So we'll see what are the different pathways in which glycolate, or in other words glyoxylate, is going to get catabolized. How do we assimilate the 2-carbon compounds? Let's look at that. These are the 3 major pathways by which glyoxylate can be catabolized, or you can say that in other words, the 2-carbon compounds are being assimilated into biomass. So if the organism is growing in the presence of these 2-carbon compounds, and if it has these pathways in it, then actually convert those compounds into its own biomass. That's what we call as assimilation. The first pathway is dicarboxylic acid cycle, which is a type of oxidative catabolism, or something that occurs during the aerobic conditions. The second one we have is glycerate pathway, where you again have certain aerobic as well as anaerobic organisms going through or taking up the glycerate pathway. And the last one is beta hydroxy aspartate pathway, which is also seen mainly in aerobic conditions. So we'll see briefly what each of these pathways mean and how are they going to help us break down the glyoxylate. Now, the organisms which carry out these glyoxylate catabolism, or which have the ability to utilize or assimilate glyoxylate or the 2-carbon compounds, are listed over here. Not all of them follow all the cycles. For example, we have Pseudomonas. Pseudomonas is known to show the aerobic form of glycerate pathway. On the other hand, we have Murella. Murella is a thermophilic acetogen. So Murella utilizes the glycerate pathway. So any other kind of 2-carbon substrates, everything is being utilized by the glycerate pathway, by Murella. If we see Oxalobacter, Oxalobacter is also utilizing the glycerate pathway, but it is an anaerobic organism. What I'm trying to tell you here is that these organisms which are listed over here don't follow the same pathway, but their main aim is to break down or to catabolize glyoxylate. So Pseudomonas is, would show you the glycerate pathway. We have the Murella also, which shows us glycerate pathway. There's Oxalobacter, which shows the glycerate pathway. Then we have the Micrococcus denitrificans, which shows the beta hydroxy acetate pathway, and E. coli can show both glycerate pathway as well as the dicarboxylic acid cycle. So let's now see what each of these pathways mean, or how is the glyoxylate being metabolized by each of these pathways. The first we have is dicarboxylic acid cycle. The dicarboxylic acid cycle is very similar to glyoxylate cycle, which is seen in many organisms. So let me first tell you what glyoxylate cycle is, and then we can see what is the difference with the dicarboxylic acid cycle. Glyoxylate cycle or dicarboxylic acid cycle is a variant of TCA cycle, that is tricarboxylic acid, or what we also call as Krebs cycle or citric acid cycle. You can find a video for that on my channel. Now the glyoxylate cycle is occurring in the peroxisomes, and here what happens is we have isocitrate. Okay, please make note, this is what I'm telling you is for the glyoxylate cycle. So isocitrate, which is a 6-carbon compound, is getting acted upon by the enzyme isocitrate lyase. This is an enzyme which is found only in the glyoxylate cycle. This splits the 6-carbon compound into glyoxylate, which is a 2-carbon compound, and we get succinate. So glyoxylate and succinate. Succinate is a 4-carbon compound. This is what is happening in the glyoxylate. Now this glyoxylate then combines with one molecule of acetyl-CoA. This is a 2-carbon compound, and it gives us by the enzyme malate synthase. This again is typical to the glyoxylate cycle. So malate synthase and isocitrate lyase are two enzymes which are found only in organisms that can bring about the glyoxylate cycle. The malate is then converted into oxaloacetate, and then the oxaloacetate gets into gluconeogenesis, basically the production of glucose. This, what I have shown you here, is the glyoxylate pathway. The dicarboxylic acid cycle is very similar to the glyoxylate pathway. Why is that? In the dicarboxylic acid cycle, as you can see, from glycolate, we get glyoxylate. So we don't have the role of isocitrate over here, but we reach glyoxylate. And then from there, things are similar. So if you see here, glyoxylate is then combining with one molecule of acetyl-CoA to form malate. So this is a 2-carbon compound, and glyoxylate is also a 2-carbon compound. So it gives us malate. The enzyme is malate synthase. Malate then gives us oxaloacetate, and oxaloacetate by various steps, gives us carbon dioxide, or it can actually form glucose. From pyruvate we can either get glucose or it gets into the complete breakdown. It differs in each pathway. But what you need to remember here is that we are trying to use a 2-carbon compound which is glycolate, and then trying to assimilate it into the body of the cell or into the body of the organism. So the glycolate here is then getting, either forming the carbon dioxide, or it is giving you pyruvate, which then forms the glucose by gluconeogenesis. So it is giving you a carbohydrate, or it can even get into the, in case of carboxylic acid, dicarboxylic acid cycle, you can even have amino acid synthesis that is taking place. So dicarboxylic acid cycle is a type of oxidative catabolism, very similar to tricarboxylic acid cycle, and that is what has been shown over here. The next one we have is the glycerate pathway. Now glycerate pathway is where you have two molecules of glyoxylate. Again, we have glycolate, which gives us glyoxylate, and then glyoxylate combines with another molecule of glyoxylate to finally give us glycerate, 2-phosphoglycerate or glycerate, which then is formed, or which then gets into, either it can form 3-phosphoglycerate and then undergo glycolysis, or it can also enter into gluconeogenesis. So glycerate pathway, if it is being used for the glyoxylate metabolism, if it is being used for production of carbohydrates, then from glycerate it undergoes gluconeogenesis, or it gets into the production of gluconeogenesis or the production of glucose. The other way is where it can get into glycolysis, that is breakdown of glucose. I have shown you breakdown, but please remember that if we are talking about glycerate pathway for production of carbohydrates, then instead of glycolysis, it gets into gluconeogenesis. So glycerate pathway is where glyoxylate molecules combine to give us phosphoglycerate, 2-phosphoglycerate and carbon dioxide. You need not remember the individual intermediates. You know what is being formed at the end. The third pathway that we have is the beta hydroxy aspartate pathway. Now, in the beta hydroxy aspartate pathway, glycolate is first oxidized into glyoxylate, same like the other pathways, and then it is converted into oxaloacetate in multiple enzymatic steps, actually 4 enzymatic steps. So let me just write it easier way instead of all the cycle that you're seeing over here. The easier form for you to remember would be from glycolate, okay, from glycolate we get glyoxylate, and then the glyoxylate combines with another molecule of glyoxylate. So I have 2 molecules of glyoxylate combining with each other to give us oxaloacetate. This is what is seen in the beta hydroxy aspartate pathway. So this pathway is actually helping us to directly form the 4 carbon compound, that's oxaloacetate, from glycolate without losing any carbon or nitrogen. That's why this is one of the most efficient pathways for glyoxylate cycles or for glyoxylate metabolism, because you're directly getting the 4 carbon compound without losing any carbon in the form of carbon dioxide. So beta hydroxy aspartate pathway gives us oxaloacetate instead of glycerate or anything else. These are the 3 pathways of glyoxylate metabolism. One is the dicarboxylic acid cycle, which is quite similar to the TCA cycle. Then we have the glycerate pathway where glyoxylate molecules combine to give us glycerate and carbon dioxide. And then we have the beta hydroxy aspartate pathway where glyoxylate molecules combine to give us oxaloacetate. These are the 3 pathways by which organisms can assimilate or utilize the 2 carbon compounds, namely glycolate and glyoxylate. So if you can see here, all the 3 pathways have been shown to you. We have the glycerate pathway as seen over here. So if you can see, we have glycolate, which gives us glyoxylate. Glyoxylate can be taken in for amino acid synthesis, that's glycine synthesis, which then later gives us serine and so on. Or you can have glycerate that's being formed. Glycerate would then, we have glycolate giving us glyoxylate and then finally giving us glycerate. This would be the glycerate pathway. Or it can get into the second pathway where we have formation of, this what is shown over here is the DCA cycle or the dicarboxylic acid cycle. Or oxalate could also combine to give us oxaloacetate, which has not been shown here, but that would be the third pathway. So these are the 3 pathways for glyoxylate catabolism or metabolism. So summing up, glyoxylate and glycolate are 2 carbon compounds. Glycolate is formed in the chloroplast as a product of photorespiration, and then it gets converted into glyoxylate by the process of oxidation. This glyoxylate can be catabolized or can be assimilated or can be used for either energy production, or it can be converted into biomass, that is, it is getting assimilated into the organism. So the end product may be different, but then the glyoxylate is being catabolized, the glyoxylate is being used up. One way it can be done is by the dicarboxylic acid cycle. The second way is by the glycerate pathway, and the third one is by the beta hydroxy acetate pathway. In all the 3, what happens is you have glycolate giving us glyoxylate, and then that glyoxylate either gives you glucose if the aim is to form a carbohydrate, or it gives you an amino acid like glycine, which then later gives you other amino acids. I hope this video was useful to all of you. See you in the next one as well. Bye.